this unit's going to be talking about cells. And before we talk about cells, let's introduce something that we'll focus on towards the end of the semester, too. That's on classification. So classification is a way of organizing all the variety of living things that there are in this world. So the broadest, biggest categories are called domains. You might thought it was kingdoms, but it's actually domains. And there's 3 domains. Okay, there's the domain that we're in. Okay, these are kingdoms up here. Protista, Plantae, Fungi, and Animalia. So these right here are kingdoms. And you probably can call this a kingdom as well, Monera, for the bacteria and the things called archaea. But these 5 kingdoms right here are what make up the domain eukarya. And eukarya basically have what we call eukaryotic cells. Eukaryotic cells. So the eukaryotic cell has a nucleus that protects the DNA. Okay, so the DNA is stored, kept, never taken out. So DNA is stored in a cell nucleus. Don't get a cell nucleus mixed up with the atomic nucleus. It's much, much bigger. So the kingdoms Protista, plant, fungi, animal, they're all part of the domain eukarya. Again, there's 3 domains. Okay, bacteria and archaea, we'll talk about that next unit. Those two domains we'll talk about in the next unit. But Monera are prokaryotes. Bacteria and archaea are prokaryotic. That means they have prokaryotic cells. And a prokaryotic cell has no cell nucleus. There's no cell nucleus. But there, of course, has to be DNA. So there's DNA, but it's not protected by a nucleus. So we're going to talk only about the eukaryotes in general, the eukaryotic cell in this unit. So this picture is going to be, it's called a phylogenetic tree. And we'll talk about this when we go to the evolution unit. So this basically shows time passing like this. So this is the present time. As you get up and down here would be prehistoric time, when life first evolved on Earth. So this common ancestor is not living anymore, but it evolved into these different domains. Okay, so looks like the first to evolve were the bacteria on Earth because you can see it branching off right there. Then the archaea later in time branched off, evolved from the bacteria. And then eukarya, the eukaryotes, evolved around the time that the archaea did. So there's two types of cells. The simpler ones, the ones that evolved first, prokaryotic cells, and then the more complicated ones, like our cells, the eukaryotic cells. Okay, let's talk about what they have in common, these two types. Well, they both have a cell membrane, a plasma membrane. Same type, different proteins, but same phospholipid bilayer. They contain a semi-fluid gel-like substance called the cytoplasm. Really, it's called the cytosol. The cytosol is the liquidy part. There's solid things in the cytoplasm, in the cytosol, and the solid things plus the liquid stuff, the cytosol, is called the cytoplasm. So cytoplasm equals the cytosol, the liquidy part, and the organelles. Organelles like ribosomes, for eukaryotes, the mitochondria, chloroplast, endoplasmic reticulum. Well, you've had these in grade school, but we'll review all that coming up soon. Okay, all cells contain chromosomes, which has the DNA in it. Okay, DNA is, like we talked about, the nucleic acid, but it's wrapped around in, it's wrapped around in a protein jacket to make these things called chromosomes, which we'll talk about later. But to be more general, all cells contain DNA, whether they're prokaryotic or eukaryotic. Okay, all cells have ribosomes. Ribosomes are the workbenches, the factories where the proteins are built from amino acids from the directions in the DNA. Okay, so ribosomes are, we'll talk about them, they're non-membrane bound, so they don't have their own membrane. Organelle is is like an organ in our body. It does a specific thing, but it's the simplest of the organelles, and it's the only organelle that prokaryotes have. Eukaryotes have a ton of other kinds. So here's a picture of prokaryotes, bacteria or archaea, and eukaryotes, eukaryotic cells. Okay, so you can see the ribosomes, these little dots right here, where the proteins are built. Okay, then you have the cytoplasm. Cytoplasm is everything within the cell, underneath the cell membrane. Okay, so the DNA are in the chromosomes, bacteria only have one chromosome, and that's that thing right there. Whereas inside the nucleus, the chromosomes, there's multiple chromosomes in the nucleus of a eukaryotic cell. Then, of course, all cells have a cell membrane. Now, there's differences here. We'll talk about those coming up. So let's talk about some general differences between prokaryotic and eukaryotic cells. Well, eukaryotic cells are much bigger than prokaryotic cells. If this was the size under a microscope of a eukaryotic cell, a bacterial cell would be about that big. Eukaryotic cells have membrane-bound organelles. That means organelles, or they call them compartments, that are membrane-bound. And in these compartments, in these spaces that are membrane-bound, specific things are done. Just like organs in our body do a specific thing. These aren't organs, they're tiny organelles. Of course, the eukaryotic cell has a membrane-bound nucleus. Well, prokaryotes do not have a nucleus in their cell. Eukaryotic cells can differentiate into different functioning cells in an organism. For example, our muscle cells, our nerve cells, our skin cells. These cells look very, very different, but yet they all have the same DNA. They have differentiated or specialized into cells that have a specific function. Prokaryotic cells don't do that. They're all the prokaryotic cells of a certain kind of bacteria or archaea function and look the same. So here you see a cell, a eukaryotic cell, compared to a prokaryotic cell. Okay, this is an animal cell. And you can see all kinds of organelles in here, different kinds of organelles. And nucleus, obviously, is an organelle. So, the size difference, notice. Bacteria on the left right here, a prokaryote, compared to eukaryote. So as the earth changed over time, the life that is found on earth also changed. So very early on, when the earth was forming, when life first existed, it consisted of single-celled organisms, such as prokaryotes. There are eukaryotes that are single-celled, but they didn't evolve, they didn't appear until 2 billion years ago. Okay, so some bacteria were photosynthetic. So photosynthesis is taking H2O and carbon dioxide and using the power of the sunlight, the energy in sunlight, to make glucose plus oxygen gas. Okay, if I balance this equation, I'd have to put 6's here. Anyway, the point is, oxygen gas is necessary for complex cells to form. So these simple photosynthetic bacteria gradually over billions of years, several billion years, build up the oxygen in the air so that eukaryotic cells, which are more complex and need more oxygen, could evolve. Then eventually, those were single-celled organisms, eukaryotes. Then eventually, about 1 billion years ago, multicellular plants evolved. And then just 660 million years ago, animals evolved. So the first eukaryotes were probably Protista. They're single cell. Kingdom Protista. Here's a protist. It looks like a little algae organism. It's one cell. You can see organelles in it. This probably is a nucleus right there. Don't know for sure. And it's green, so it's photosynthetic. You might say, why did life evolve to be multicellular? Why couldn't a cell just grow bigger and bigger and bigger? And that would be the organism with more compartments in it. And why are all eukaryotic organisms consisting of these tiny eukaryotic cells? Well, a cell size is limited by its surface area to volume ratio. Okay, so surface area is like if you had a cell and you had to paint the outside. The outside of the cell is the surface area. The volume is the space it takes up inside the cell. Now, as the cell gets bigger, the surface area doesn't increase as much as the volume does. So the volume increases at a greater rate than the surface area does. And this poses a problem. Surface area is where you bring in nutrients, oxygen, the cell membrane, and get rid of waste products. So if the inside of the cell is getting bigger and bigger and you can't get enough food and oxygen inside the cell for that big volume inside the cell, and get rid of your waste products from inside the cell to outside the cell, that's going to cause a problem because the cell won't be able to function properly. It's not getting rid of its waste products fast enough for the size it is, and it's not getting enough food and oxygen. So there's a limit to what a cell size can be. So again, as it grows, the surface area gets bigger, but the volume gets much bigger. So the surface area to volume ratio actually would decrease with increasing cell size. And that's not good. So that's why we don't have big, gigantic cells. But we're made of millions of cells instead of being made of a few big cells. So here you see some actual numbers for a cubical size cell. Surface area, the volume, and then the ratio, surface area to volume ratio. You can see how as the cell grows, left to right, both of these numbers are increasing. Okay? But the volume is increasing at a much bigger rate than the surface area. Just look at it. 864, 216, 512. Compare this to the rise in the surface area, 24, 96, 216, 384. It's not increasing as fast as the volume is. So if you divide the surface area number by the volume number, you'll get this ratio, and they did the work for you. And you can see that this ratio is getting smaller with the bigger the cell gets as it grows. So the problem is, as you get bigger, the surface area to volume ratio decreases. And even though you can do more things faster because you got more volume to work, you're not supplying enough nutrients and oxygen, and you're not getting rid of the waste products, so it's not an efficient process. So what a cell does when it gets to a certain size, instead of getting bigger and bigger, it divides into smaller cells that then will grow. And so that's why we're composed of millions of cells rather than 100 big cells. A eukaryotic cell is bigger than a prokaryotic cell because it increases its surface area by making these compartments inside. And those compartments are called organelles, membrane-bound compartments. Each of these different types of organelles has a specific function. So don't confuse an organelle with tissue or an organ. It's not. But does a specific thing like the organs in our body, like our heart is made of thousands of cells, probably millions of cells, but yet, and it does a specific function, pumping blood, but organelles are just a little piece of a cell, not many cells like an organ is. The same kind of concept. They have a specific job to do, organelles and organs. So if I give you a picture of a cell, here's a eukaryotic cell and here's a prokaryotic cell. They kind of just they put them on the same, next to each other to compare them. You should know all these in yellow I highlighted in yellow the organelles. So you should know their function and identify them in a picture. Okay, here we have an animal cell showing cut away, showing some of the organelles. Okay, let's start here. The nucleus is this big purple thing. I'll save the details for later. Then that connected to the nuclear membrane. So it's called the nuclear membrane. Not just a nuclear envelope. I like to call it nuclear membrane. Okay, surrounded by that is the rough ER. So RER. Now, the rough ER is hard to see here. It's speckled with ribosomes. Okay, so they're showing the little speckles with those two arrows. Now, ribosomes are also free-floating in the cytoplasm, like right here, as you can see, all throughout, actually. Okay, then the rough ER is attached, turns into smooth ER. There's no ribosomes on it. Like there is on the rough ER. And then in the cytoplasm, which is between the in the cytoplasm, which is inside the cell membrane, all this blue stuff containing the organelles as well. We got mitochondria. Plasma membrane is the cell membrane, right? We got mitochondria that look about the size of a bacteria. Those are where our food actually, the energy in our food is extracted and turned into ATP. So these are the powerhouses of the cell. Is where its energy is released by breakdown of food using oxygen. Much more on that later. So they're like the powerhouses of the cell. They provide energy in the form of ATP. Okay. Microtubules. Okay, all throughout the cytoplasm are protein fibers that impart a major function of protein fibers inside the cytoplasm is to be the cytoskeleton, where the organelles are actually supported and hooked up to. Then we have the centrioles. This organelle actually spews out microtubules and forms what is called the spindle when cell division is occurring, and the microtubule fibers in the spindle connect to the chromosomes and then reels them in so that each side of the cell, each new cell, gets the same number of chromosomes and same type of chromosomes. More on that later. Lysosomes are membrane-bound sacs of digestive enzymes. It's like the demolition crew in that the digestive enzymes will break down any molecules that aren't that aren't needed anymore. Or structures. Maybe it'll break down broken organelles. Those digest enzymes do that inside the lysosome. Okay, then we have a secretory vesicle. Those vesicles pinch off this thing, the Golgi. Okay, it pinches off the Golgi. And they're called secretory vesicles because the cell will secrete them with the proteins that were made on ribosomes or in the rough ER. It will secrete those proteins in a little membrane-bound sac that comes off the cell membrane, pinches off the cell membrane. Okay. So we talk about the Golgi apparatus. Looks like a stack of pancakes. Okay, we'll talk more about all that coming up. Then we have peroxisome. Peroxisome contains hydrogen peroxide. And that breaks down free radicals, which are chemicals when you digest something. The free radicals can hurt the cell. So these are little chemical groups that are reactive and can damage a cell. So the peroxisome uses hydrogen peroxide to neutralize, to react with those free radicals. So which of the following are prokaryotic cells? Well, bacteria and eubacteria. Bacteria, either eubacteria or archaea, either one. Or you could say Monera. What are the domains of life? Well, there's 3: archaea, bacteria, sometimes called eubacteria, like I just said before. Archaea and eukaryotes. Okay, so there we go, right there. This one, letter D. What are the kingdoms of eukaryotic? Okay, so protists, fungi, plants, and animals. There you go. Which is not a basic feature of all cells. All cells are surrounded by a plasma membrane. That is a basic feature, so don't pick it. All cells contain a semi-fluid substance called the cytoplasm. Well, really it's called the cytosol, but it's good enough. So all cells have that, so good. That can't be the right answer because they want the one that isn't a feature of all cells. All cells contain structures called chromosomes, so far so good, which are contained in the cell nucleus. No, that is not true of all cells, only true of eukaryotic cells. So I'm going to mark that as right. All cells have ribosomes, indeed they do. So that's not right. All eukaryotes are multicellular. No. They're single-cell protists. Even yeast, a fungi, is unicellular. Uni meaning one. So here's a picture of amoeba is animal-like protist. That's one cell. Euglena is a photosynthetic, a plant-like protist. And Paramecium is an animal-like protist in the way that he doesn't make his food etc. He has to eat other living things. But all three of these are protists, and these two are animal-like protists, and this guy is a plant-like protist, making his own food with sunlight. So, which one of these characteristics is not a eukaryotic characteristic? They may be multicellular. Yeah, sure, that's true. They may be single-celled. Well, that's true too. Cells have compartments called organelles. Well, that's true. I would say membrane-bound. I wouldn't call a ribosome a compartment. But anyway, their cells lack a nucleus. No, eukaryotes have a nucleus. So, lack a cell nucleus, of course. So that's the correct answer right there. Okay, so what do all cells have in common? Well, prokaryotes have ribosomes, which are organelles, so I'll keep that one. They all have cytoplasm. They don't all have a nucleus, and they don't all have a large size. So they should have really changed this answer to membrane-bound organelles. And so eukaryotes have those, but prokaryotes don't. Both of them have cytoplasm. So cytoplasm would be the correct answer. Okay, so we're supposed to rank these things from size and complexity. So from smallest amount to largest. So the smallest would be atoms. So right there, and the biggest of these would be cells. So I pick letter D. How did eukaryotes solve the problem of small surface area to volume ratio as they grew bigger? Well, by using organelles. Also by dividing, doing cell division, but so did bacteria. So that's why eukaryotes are significantly bigger than prokaryotes. So please, again, don't get mixed up with the atomic nucleus compared to the cell nucleus. Cell nucleus is many times bigger than an atom. It consists of millions of atoms. Billions of atoms, probably. The nucleus is called the control center. I like to think of it as the bank vault that safeguards, that protects and stores all the protein recipes that the cell needs in code form in the DNA molecule. So we call that a prokaryote. Bacteria, or archaea, they have no nucleus, whereas eukaryote has a true nucleus, a cell nucleus. Okay, so here's the cell. Here's the nucleus right here. Inside nucleus is this Well, inside the nucleus are chromosomes or chromatid or chromatin. Chromosomes, that X thing shown right there in the picture is a chromosome, is DNA, very coiled up. And wrapped in protein. So you can see it on a microscope, but you only see chromosomes when the cell is dividing. You don't see chromosomes in the nucleus unless the cell is undergoing cell division. Okay, so usually it's in chromatin. The DNA is in chromatin form, and that's a loose form of DNA, and it's diffused, so you don't see it under a microscope. So what you see in that picture is a chromosome, but chromatin is like soupy. I call it soupy DNA. The DNA is not all tightly wound up and associated with lots of protein. It has some protein on it still, but basically it's loose. Why is it loose? Well, it's when the cell is not dividing. It has to be available. The recipes have to be available so that the cell can make a certain protein at any time by reading the code on the loose DNA. When the DNA is all tangled up in a chromosome, you can't access the recipes. This is to remind you that prokaryotes have one chromosome, which is like one chromosome of DNA. DNA is double-stranded, of course, and the chromosome is circular, even though it's all tangled up. Now, eukaryotes have, depending on what type of organism it is, depends on how many chromosomes the eukaryotic cell has. So we have 46 chromosomes, or 23 pairs. A fern, this species of fern has 1,440 chromosomes, or what would be at 720 pairs of chromosomes. So it varies how many chromosomes a eukaryotic cell has depending on the species. Now, chromosomes are made of tightly coiled DNA and histone proteins. Histone proteins combine and wind up the DNA. Now, chromatin, when this is poorly written, let's put a period right there and cross out cog, because you don't call chromosomes chromatin. Okay, chromatin is DNA and histone proteins, but not tightly wound. Actually, loose. So I call it soupy DNA. So this picture is showing stages of DNA getting wrapped into fibers that make up the chromosome. So histone are the proteins. When there's only a few of those histones on the DNA, it's chromatin. They're talking about this being a chromatin fiber. Not really. That is, I guess you can call it that, but a very, very tightly wound chromatin fiber that makes up that chromosome that you saw on the other page. Okay, this is kind of confusing. These two, there's one here, one right here. Okay, so these are called chromatids. So when you have these two branches are called chromatids or sister chromatids because they're on the same chromosome. That's important to know. Okay, the two chromatids are bonded together by a centromere. That's kind of easy to remember, centromere. And the telomere is just a region on the ends of the chromatid. As we age, even though we keep on making new cells, those chromosomes start to wear at the telomeres. And so once the telomeres start wearing, some crucial protein recipes will start being damaged. And that's part of the aging process, which eventually, terminates with death. So nucleus has a membrane around it, and it's kind of a special membrane. It's called the nuclear membrane or the nuclear envelope. I always call it the nuclear membrane. Okay, but the weird thing about that, several weird things, is it's a double cell membrane. So it's not just a lipid bilayer, it's two lipid bilayers. So there's 4 layers of phospholipids. Okay, that's important. There's also nuclear pores through this double membrane. You can see one right here. Again, this is the nucleus, not a cell. Okay, so there's a region that stains clearly for RNA in the nucleus, and it's called the nucleolus. And that's where ribosomal RNA is made, and the ribosomes are built. And then they're passed through the pore to the cytoplasm or to the rough ER. Okay, right in this picture, all this soupy stuff right here is chromatin. You don't see the chromosomes condensed with their nucleotides all wound up when the cell's not in cell division. The DNA is loose, so you're not going to be able to see it with a microscope. So, major function of the nucleus is to be a safe for all the DNA that codes for proteins that the cell might need. Also, they assemble the ribosomes in a nucleolus, and it's just not our RNA that is copied off the DNA. It's all the other kinds of DNA that are needed to make proteins. So remember, there's other kinds. There's tRNA and messenger RNA. So all three of these RNAs are needed for the process of decoding the DNA instructions, the genes, into proteins. We'll talk about that in another unit coming up. Okay, cells that can contain a true nucleus and other membrane-bound organelles obviously are eukaryotes. Where is the DNA of a eukaryote found? Of course, in the nucleus. You say in the nucleolus too. Okay, but nucleus is the more straightforward answer. How does the nucleus control the activity of the cell? By making DNA, by directing protein synthesis, by allowing DNA to leave the nucleus. All that does not happen. By to understand instructions of the mitochondria. So it's between A and B. Now, we haven't really talked about this yet, but in the nucleus, before cell division, the DNA has to be copied. But I don't think that controls the activities of the cell. So, that's what happens in the nucleus. I would cut it out because it's not controlling anything. By directing protein synthesis, making proteins. Well, DNA makes in the nucleus the RNA that carries out protein synthesis, whether it's mRNA, tRNA, or rRNA, that RNA directs the protein synthesis. So, because that RNA is made of the DNA codes, ultimately, the nucleus controls protein synthesis, the decoding of the DNA to make protein. So I say B. So details on this is going to be given later, but the messenger RNA is a copy of a certain portion of the DNA, the gene. The copying of the gene into mRNA inside the nucleus is called transcription. Transcription. Okay, that's where DNA is turned into RNA, messenger RNA, or tRNA, or RNA. But the messenger RNA is the copy of the gene to make a protein. Okay, so once the messenger RNA is made, it has to exit the nucleus through the nuclear pores. And then it's gonna do all kinds of other stuff, technical stuff that we'll talk about in another unit in order to make proteins. But it will happen on the ribosome in the cytoplasm. Remember that ribosome is the workbench on which the protein is snapped together from amino acids following the code on the messenger RNA. Okay, we can say that the cell, like our body, has a system, like our digestive system is a bunch of organs acting together. For example, our mouth, our teeth, our salivary glands, the stomach, the small intestines, large intestine, they all are basically working together. Each one of those parts of the digestive system doing a specific thing, but together working as a system. So in a cell, we have a system called the endomembrane system, and it consists of the nuclear membrane, which is connected to the rough ER, which is then connected to the smooth ER, and then it's not connected to Golgi, but vesicles from the smooth ER will travel to the Golgi. So basically, it's in close communication with the endoplasmic reticulum. And then the Golgi pinches up transport vesicles, and some of those transport vesicles are lysosomes, and some of those transport vessels contain proteins that are used by the cell, and they're not secreted, and some are secreted at the cell membrane. So I would differ with this. The Golgi is not directly attached to the endoplasmic reticulum, but the smooth ER sends out vesicles to the Golgi through the cytoplasm. Here's a good picture of showing the nuclear envelope and how it's connected to the rough ER, which is right here, RER, with the little dots, the ribosomes on them. Then the rough ER is connected or turns into the smooth ER, which you see right here. And these places are, well, the rough ERs were proteins on the ribosomes that are going to be used by, that are going to be exported by the cell, are made. So again, the ribosomes on the rough ER are where proteins will be built, but proteins that will be secreted or put into the lysosome. Okay, that's kind of important. The ribosomes outside, which aren't part of the endoplasmic reticulum, will build the proteins that the cell itself needs, except for the lysosome ones. And those will be used by the cell, not secreted. Okay, so the smooth ER sends out little blobs of vesicles. Well, that one doesn't look like this one right here. It blobs off of the smooth ER. I always spell vesicle wrong. There's only one S in vesicle. Okay, so this is the vesicle from the smooth ER. You can see one forming here. Okay, and that's going to go and combine, fuse with the Golgi apparatus. Okay, the Golgi apparatus makes some changes to the protein inside that vesicle that fused with it, and then it secretes its own little blobs, its own secretory vesicles that then go to the membrane, unless they're lysosomes, and are exported, the contents being exported into the extracellular environment, the liquid outside the cell. Here's a better picture of the nuclear envelope, the rough ER, and the smooth ER. Okay, these are basically membranous tunnels, like subway tunnels, that go throughout the cell. Okay, so this is the rough ER, part of the rough ER. Here's a ribosome. A protein will be built on that ribosome and enters the inside of the RER, the inner compartment, which is called the space inside any kind of tube. It's called the lumen. So here's a protein. This is a ribosome. But the protein will be built and inserted into the rough ER. Okay, in the rough ER, those new proteins are trimmed up and folded according to the types of amino acids that are in that protein. As we talked about, they're given their tertiary structure. So here you see a ribosome. They're showing it right there. This guy right there, the gray thing right there, is messenger RNA. Messenger RNA is being read. This is called translation. We'll get to that later. When the mRNA is read, and the protein is inserted, the new protein, this thing's a protein, the protein being formed is this thing. And what happens is mRNA feeds through the ribosome. The code is read. And those are instructions for snapping together the right amino acids to make this protein right there. The protein is inserted into the lumen for further processing. This is the inside of the rough ER. And or it's inserted into the membrane and becomes one of the membrane proteins. Okay, let's talk about ribosomes. Ribosomes are made of rRNA, ribosomal RNA, which is made in the nucleolus. Remember that? And protein. The rRNA and the proteins, not histones, other kind of proteins that are already there in the cytoplasm, complex together and make the ribosome. Now, so there's a little slot here, right here, where the messenger RNA can go through, can slide through and get decoded by the ribosome. A ribosome consists of two subunits. The large subunit, which is this gray thing up here, and the small subunit. And there's a groove between them where the messenger RNA goes in. Okay, so the mRNA goes through like a train on a railroad track. And as it passes through, the ribosome reads off the code, the genetic code, which the messenger RNA is a copy of one of the genes from the DNA. This is called translation. When mRNA turns into protein. So let's get this straight. In the nucleus, DNA, parts of the DNA, is turned into messenger RNA. Sorry, that's called transcription. And then, well, it could be also. It could turn into RNA and tRNA, which I won't mention yet. But when DNA turns into RNA, this is called transcription. Then the messenger RNA will be transcribed into protein. This happens in the cytoplasm. And that's called translation. Translating from the RNA language to the protein language. So you kind of need to know, be aware of, be aware of these two terms. Okay, so here's some review questions. Where is the ribosomal RNA in the ribosomal subunits made? What location is rRNA manufactured? That's nucleolus. You could say nucleus, but the more specific answer is nucleolus. What do ribosomes consist of? Well, obviously rRNA and proteins, but not histones. So there we go. This all the parts of the endomembrane system. Rough and smooth ER, yes. Golgi, yes. Apparatus, I just call it Golgi. Lysosomes, yes. So this is it. Well, so is that. Nucleus, it's not the nucleus, it's the nuclear membrane. So this is almost right, but I wouldn't pick that because it's supposed to be the nuclear membrane. Nucleus, again, no. Okay, this could be a little tricky question. Where are proteins made? Well, they're made on the ribosome, so that's the most obvious question. Which is now smooth ER does not make proteins. It customizes them. It puts the finishing touches on them, but it doesn't make proteins. So it's not the smooth ER. DNA, well, it's involved because that's where the messenger RNA is. We just said the nucleus is the control center. So obviously, it's important for making proteins, but the most direct answer where proteins are actually being snapped together, the amino acids are being snapped together to make proteins, that's on or in the ribosome. DNA, so nuclear membrane, yeah, I mean, if the nuclear membrane has holes in it so that the mRNA made in the nucleus can go into the cytoplasm, but it's not as direct as ribosomes. Okay, the smooth ER, remember, is connected to the rough ER, and it will take those proteins from the rough ER and put them in membrane-bound packages and secrete them into the cytoplasm, where they'll go to the Golgi for further processing. Okay, so it exports vesicles with proteins to the Golgi, where those vesicles fuse with the Golgi and release their contents. But primarily, the smoothie R makes lipids, primarily steroids, primarily cholesterol, and steroid hormones, which are based on the cholesterol structural formula. They also have important, in certain tissues, they have important detoxification function. They break down inside the smoothie R. They break down the poisons or drugs that can harm the cell. And special in muscle cells, calcium ions are very important for contraction. So special kinds of smoothie are there to store calcium ions. But you don't need to know that. So membrane proteins with carbohydrate chains connected to the protein are called glycoproteins. And these are, the sugar part is attached to the protein part in the rough ER and in the Golgi. Okay, the Golgi will trim the sugar part, the sugar chain of the glycoprotein. It will trim it up so that when it secretes it on a vesicle, this is like a dress label, and it will head on over to where it's needed on the cell membrane, or maybe it's the membrane of another organelle like a mitochondria or a chloroplast. Okay, so the Golgi is modifies that sugar molecule part of the glycoprotein. That sugar chain on the protein acts like a zip code that tells the protein where it should go after the vesicle is exported by the Golgi. So this picture shows the smoothie R exporting a vesicle of protein, or it could be a vesicle of lipids, steroids, cholesterol, and that vesicle travels along the cytoskeleton microtubules to the Golgi, and that membrane will fuse with the membrane. The vesicle membrane will fuse with the membrane of the Golgi apparatus and dump its contents into the Golgi, which will process it and then butt off another vesicle with the finished product that then goes to the cell membrane for export out of the cell. Or it could be exported to another organelle membrane and become incorporated on that. Okay, so we said the Golgi is like a post office in sorting the mail, putting the right tags on it, the glycolipid I'm sorry, the glycolipid or the glycoprotein on on the protein mating, the rough ER, and then sending it out. So here's the transport vesicle from the smooth ER, blending in with the fusing onto the Golgi giving its contents to the inside of the Golgi, the lumen of the Golgi, and then the Golgi processing it, and then forming new vesicles that are transported out of the cell or to other organelles inside the cell. So these vesicles that the Golgi exports will move to lysosomes. No, they'll be lysosomes if their contents are the digestive enzymes, or they'll be inserted into the cell membrane of that cell, or they'll be exported. The vesicle will fuse with the cell membrane and release its contents to the fluid outside the cell. Okay, this picture is showing that the rough ER makes this is RER, makes a lysosome. And the lysosome could be imported into the Golgi and then labeled, zip coded, so that it's directed to a vacuole, a food vacuole is just the membrane opening up and taking in a big chunk of food. Big compared to molecules. And so the lysosome will fuse with that food vacuole, and digestion will occur in that. The molecules will be broken down, and the monomers will be released into the cytoplasm. Or the Golgi can make its own big lysosome that will be targeted to an old or damaged organelle, and it will digest that damaged organelle in the cell, breaking down into monomers, and the monomers will be released into the cytoplasm. Okay, the endomembrane system serves to shift cell products to places in a cell. Well, that makes sense. No, nothing to do with DNA. No, it doesn't give directions. It doesn't direct anything. No, that's the cytoskeleton, letter D. So it has to be A to export cell products, proteins, basically proteins, and lipids to places in the cell. This is easy. Rough ER has ribosomes. Smooth ER doesn't. Where in the cell are lipids made? Okay, that would be the SER. Smooth endoplasmic reticulum. Okay, this is backwards. The Golgi takes the vesicles from the smooth ER to transport. Okay, so that's wrong. Okay, the sacs making the Golgi are not connected. No, that's true. They look like flattened pancakes stacked up on each other. They send vesicles to each other, yeah, but they're not connected. So it's B. The Golgi has ribosomes? No, it doesn't. No. Okay, what modifies and receives vesicles from the endoplasm? Well, from the smooth endoplasmic reticulum is the Golgi. Sorry, I worked this out before I pushed the button. What is not a function of the lysosome? Aiding a cell in creating ribosomes? No. Lysosomes do not create anything. They store digestive enzymes. So fusing with vacuoles to digest food, yeah, that's possible. Breaking polymers into monomers, of course, that's what digestion is. Recycling worn out cell parts, yep, it's like a recycling salvage company. So A is the answer. Okay, here's another picture of the cell membrane. I don't really know why I put it here, but except to say that these proteins in the cell membrane got there from vesicles secreted either by the rough ER or secreted by the rough ER, the smooth ER, or the Golgi. So this is how a vesicle exports its contents to the outside of the cell from the inside of the cell. Okay, so this is a secretory vesicle, either from the Golgi or from the smooth ER. Okay, so what happens is the phospholipid membrane fuses with the cell membrane, and it just like dumps out the content. These are the new proteins or lipids. It could be steroids if it comes from the smooth ER. I should have mentioned this when I was talking about active and passive transport, but this exocytosis process is active transport. It requires ATP in order for this to happen. It doesn't just spontaneously happen. So exocytosis is not passive. It requires ATP as active transport. Okay, so endocytosis is the reverse of exocytosis. So that's when you import stuff like food molecules into the cell, not through a protein, but by a big vesicle. Vesicle is just a membrane-bound compartment using the cell membrane as the new membrane of the vesicle. Again, this is an example of active transport. This doesn't just happen spontaneously. It requires energy. Okay, endo means taken in. Exo means exporting, pushing out of the cell. We can be particular and talk about different types of endocytosis. If the particle is big that you're bringing in, since it's endo, a big part of the nucleus will grow out and surround the food particle and bring it in. This is what our white blood cells do when they eat germs and then they destroy them with lysosomes. Okay, this is how amoeba eats. He finds a little protozoan, or algae, and he surrounds it with his pseudopodia, false feet, and takes it in as a vacuole, then sends lysosomes to it to digest it and get his food from it. So that's called phago, means eating. Phagocytosis is a type of endocytosis. All these require ATP. All these are active transport. Now, if you just are taking in molecules, a bunch of molecules, and you want to do it quick without individual ones going through a channel protein, you just grab that concentrated extracellular fluid with the molecules that you want, and you bring it in. Those vesicles are much smaller than phagocytosis vesicles, though it doesn't look like it. So when you drink, when the membrane actually brings in nutrient-rich molecules dissolved in water, is called pinocytosis, means drinking. Now, if you want to be really, really specific of what you're bringing in, you got receptor proteins that specifically bind to the molecule, a specific molecule you want to bring into the cell. That could be a specific hormone that has a specific receptor that pinpoints the hormone and brings it in. That's called receptor-mediated endocytosis. You need a receptor protein on the surface of the membrane ready to pick up the hormone or other molecule that's out there that the cell needs. So that's just a summary of what I just talked about, 3 types of endocytosis. Now recall that all these are active transport mechanisms. When the cell brings in through a cell membrane large solid particles, this is called phagocytosis. When you have vesicles that move through the membrane because of receptor proteins, that's called receptor-mediated endocytosis. After the vesicle becomes, the membrane of the cell, the membrane of the vesicle becomes part of the plasma membrane, which I just call the cell membrane.